Hello, good day. This is the second part of our report about neurolation. And in this part, we will talk about what neural crest is, the formation of neural crest, the migration of neural crest, and the different types of neural crest. So first, what is neural crest? Neural crest produces neural crest cells which originate from the neural folds through the interaction of the neural plate with the presumptive epidermis. These neural crest cells are temporary group of cells induced at the border of the neural plate that subsequently migrate throughout the embryo and later differentiate into multiple cell types that contributes to peripheral nervous system, craniofacial cartilage and bones, skin pigment, and endocrine. To make it simple, neural crest cells become multiple different cell types and contributes to tissues and organs as an embryo develops. Let us talk about the formation of neural crest cell. Formation of the neural crest. As the neural folds elevate, cells of the lateral border or crest of the neuroepithelium, the natural crest undergoes an epithelia-mesenchymal transition, or the process by which epithelial cells lose their polarity or adhesion. So the process of formation of a neural crest is called epithelia-mesenchymal transition. Migration of the neural crest. This is the pathway by which the neural crest cells leave the neural tube depends on the region by changing their shape and other properties from those of the typical neuroepithelial cells to those of the mesenchymal cells. It also gives rise to the heterogeneous array of cells and tissues. Also, it contributes to the craniofacial skeleton and other mesenchymal derivatives. In this table, we can see the major derivatives or the portion of the cranial and sympharyngeal neural crest. Neural crest migrates first from the sensory nervous system up to the muscles. So in this illustration, we can see the migratory paths of the neural crest in the head region. Cells leaving the neural crest migrates and form structures in the face and neck. The numbers 1 to 6 are the pharyngeal arches. And at the middle part, the Roman numerals are called the epibranchial placode, or the thickenings that give rise to the sensory neurons of the peripheral nervous system. After the migration of the neural crest cell, let us go now to the types or the major derivatives of the neural crest. First is the anterior neural crest or the cranial neural crest. Anterior neural crest is the major component of the developing cephalic end of the embryo. It gives rise to the majority of the head connective and skeletal structures, nerves, and pigment cells. Also, anterior or the cranial neural crest cells differentiate into a wide variety of cell and tissues types. In this part of neural crest, it more focuses on the head part of the embryo and also the sensory nerves. Second is the circumpharyngeal neural crest. The circumpharyngeal neural crest arises in the posterior rhombencephalic region in the lower part of the pharynx. Rhombencephalic or the rhombencephalon is the posterior portion of the brain including the cerebellum and brainstem. Also, it is the neural structure of the part of the nervous system. Part of the circumpharyngeal neural crest is the vagal neural crest cells. Vagal neural crest cells migrates into the developing guts as precursors of the parasympathetic neurons of the digestive tract. It arises within the neural tube between the cranial and the trunk regions of the body axis. Also, it gives rise to the rich variety of specialized tissues and cell types throughout the body. Another part of circumpharyngeal neural crest is the cardiac neural crest cells. Cardiac neural crest cells contributes massively to the truncoconal fold. These cells considerably contributes to the heart development or the development of other organs. Several severe abnormalities of these organs results from the deficiencies of the cardiac neural crest. One such is the DiGeorge syndrome, caused by a deletion on chromosome 22 in humans. This syndrome is characterized by hypoplasia and reduced function of the thymus, hydroid, and the parathyroid glands, as well as the cardiovascular defects such as persistent truncus asterius and abnormalities of the aortic arches. Also, circumferential neural crest cells also migrate ventrally to the phalanx, and also it contributes to the connective tissues of these muscles, such as the muscles of the tongue. Any disturbance in this region of cardiac neural crest cells can result in cardiac septal defects as well as glandular and craniofacial malfunctions and malformations. The trunk neural crest. The trunk neural crest extends from the sixth somite to the most posterior somite. In embryology, somite is one of the longitudinal series of block-like segments in the middle layer of the tissue. The term somite is also used more generally to refer to the body segment of an animal. And in the trunk neural crest, there are three main migratory pathways of neural crest cells can be discerned as the dorsolateral pathway, second is the ventromedial pathway, and the third is the ventrolateral pathway. Dorsolateral pathway is a pathway where the cells go and disperse beneath the ectoderm and ultimately enter as a pigment cell. Second is the ventromedial pathway. Cells that uses this branch belong to the sympathoadrenal lineage and contribute to the sympathetic nervous system and to the adrenal medulla. Sympathoadrenal lineage means that it has already passed a number of restriction points, so they can no longer form sensory neurons, glia, or melanocytes. Lastly, the third is the ventrolateral pathway. Cells that follow this pathway form the segmentally arranged sensory or spinal ganglia. Spinal ganglia is a cluster of neurons. We must also consider the four types of cellular progeny rise from the sympadoadrenal progenitor cells. First is the molpoadrenal chromaffin cells. Second is the small, intensely fluorescent cells found in the sympathetic ganglia. Third is the adrenergic sympathetic neurons. And lastly, aspulation of cholinergic sympathetic neurons. In this table, we can see the major derivatives of the trunk neural crest. The phylogenetic origin of the neural crest. Phylogenetics is the study of evolutionary relationship among biological entities. When we say neural crest, it is a typical vertebrate feature. Its derivatives are often regarded as being homologous with the epidermal and visceral nerve plexus found in the non-vertebrates. That is why it is assumed that neural crests arouse as a result of centralization of the nervous system only seen in vertebrates. Also, neural crest cells are a transient embryonic progenitor population that emerges from the dorsal neural tube or during the early development. This is the big concept of the neural crest. So in this illustration, we can see where the major derivatives of neural crest are located, how do the migration of neural crest occurs, and the main contributions of the major derivatives of neural crest. To sum it up, we must remember the major derivatives of neural crest, and it is the anterior or cranial neural crest, second is the circumpharyngeal neural crest, and lastly, the trunk neural crest. And under the circumpharyngeal crest, there is two types of neural crest cells, which is the cardiac and vagal neural crest cells. And in the trunk neural crest cells, there are three major migratory pathways, which are the dorsolateral pathway, ventromedial pathway, and ventrolateral pathway.